Hi guys, welcome to our second lecture on muscle physiology. Today we're going to talk about skeletal muscle contraction as it's initiated at the neuromuscular junction. And we've looked at some of the features of the neuromuscular junction prior to this, and so let's review those. As a warning, make sure that you're familiar with that general myocyte anatomy, just so that you can walk through this. We're going to get into some heady stuff. So remember that the neuromuscular junction is where the neuron and the muscle meet, and neurons that innervate muscle are called motor neurons. And so here's the axon of a motor neuron. It's a myelinated motor neuron. This myelin is made up of these cells that actually act as insulators. Anyway, an electrical impulse called the action potential is propagated down the axon to this structure called the terminal bouton, all right? So the terminal bouton is the end of the axon. And you're going to notice there are these little structures in here called secretory vesicles, okay? They're vesicles that secrete, and they secrete specifically neurotransmitters. Now at the neuromuscular junction, the neurotransmitter is an excitatory neurotransmitter called acetylcholine, abbreviated ACH, acetylcholine. And acetylcholine is released from these vesicles in response to this electrical potential. Now, that electrical potential is what we call a depolarization event. So if you'll recollect from earlier in your science courses in biology, you learned about something called the sodium-potassium pump. And nerve tissue is where you find this structure. And the sodium-potassium pump, as you'll recollect, actively transports sodium ions outside to the cell and actively transports potassium to the interior of the cell. And so we say that those cells become polarized. They're polarized because I have lots of sodium outside the cell and lots of potassium inside the cell. And so I have two poles, right, where there's lots of potassium and lots of sodium. Of course, sodium wants to get in and potassium wants to get out, but they can't. They're blocked until the wave of depolarization occurs, all right? So the electrical impulse is, in fact, simply the movement of those ions into and out of the cell, respectively, as channels open. Now, when that wave of depolarization, that electrical current, reaches the axon's terminal bouton, it actually opens up some voltage-gated calcium ion channels here, okay? And they allow calcium to flow in, and that causes this secretory or vesicle fusion, releasing acetylcholine. Acetylcholine diffuses across this gap called the synapse, and it causes a depolarization in this structure called the motor end plate. I know that's a lot. Let's take a look at a static image. So again, the neuromuscular junctions where a motor neuron, in this case a myelinated motor neuron, this structure, this myelin is an insulator. This is the axon of a motor neuron. Notice that there's potentially multiple terminal boutons, but we're just going to look at this one here. All right, so an action potential, an electrical impulse, a depolarization wave, travels down the axon. When it reaches the terminal bouton, it opens up voltage-gated calcium ion channels. Now, calcium floods into this terminal bouton, and it causes these secretory vesicles to fuse to this, what we call a presynaptic membrane. Now, those secretory vesicles are filled with neurotransmitters, specifically acetylcholine. It's an excitatory neurotransmitter. And as acetylcholine is released into this synapse, this gaps, it diffuses naturally across the synapse and it binds its targets, which are receptor proteins on the postsynaptic membrane. In this case, we call that the motor end plate, right? Because this is a neuromuscular junction, right? So acetylcholine diffuses across, it binds these receptors, but these receptors are unique. They have a twofold role. They're not only receptors for acetylcholine, but they're sodium ion gated channels. Normally they're closed until acetylcholine comes and binds to them and opens it up. Now, that allows a rush of sodium into the motor end plate. A rush of sodium into the motor end plate. And in the previous illustration, you saw that we saw this flash, and that's the representation of this depolarization wave. You know, notice way over here, hopefully you can recognize this as these transverse tubules, the transverse tubules, all right? So once again, action potential, an electrical signal travels down the motor neuron, causes calcium channels to open, vesicle fusion, acetylcholine diffuses across, binds sodium ion receptors, which causes a depolarization wave in the motor end plate. So here's kind of a list of those terms. What did I miss? Terminal Butan, neurotransmitter, vesicle fusion, sodium gated channels, motor end plate depolarization. I think that's pretty good. Look, before we go on, when we talk about axons and motor neurons, we talk about motor units, right? So if you're an athlete, one of the things you want to do is create muscle memory. And the way that you do that is by repetitive motion, okay? And in these repetitive motions, you're going to activate with some sort of like a swing thought. If you're a golfer like Tiger Woods, you have this specific idea about which muscles to activate, and it'll pass down a motor neuron. Now that motor neuron, again, can have multiple axonal projections, activating multiple muscle fibers, right? But you create a memory here, a strengthened synaptic connection between this neuron and that thought in your frontal cortex, okay? And so, there are nerves in the frontal cortex of the cerebrum called motor neurons. They descend down the spinal column, and when they get to the appropriate level of your vertebral column, they synapse on a secondary motor neuron in what we call the ventral horn of the spinal cord. So this is a dissection through the spinal cord. You notice it's got this gray area in the interior. So those are cell bodies of motor neurons. So I have a neuron from my brain down to here, and then I have this secondary motor neuron that goes out and synapses onto my muscle fibers. All right, so how does it all work? Remember that acetylcholine is excitatory. When it binds to its receptor proteins, it causes these gated sodium ion channels to open up. Now that causes a wave of depolarization. All right, so remember from our previous understanding or study of the physiology or the structures of the myocyte, that there are these important structures, right? I have this like highly elaborated sarcoplasmic reticulum, but then I have these things called transverse tubules or T-tubules. Now they're actually opened up to the outside of the cell, to the extracellular space where there's lots of calcium ions, right? Remember that calcium ions flooded into the terminal Butan when they were allowed to, and they can flood in here. In fact, the depolarization wave that passes from those sodium gated channels at the motor end plate come to the transverse tubules, and they cause calcium voltage gated or depolarization gated channels to open here. All right, so the T-tubules are lined with calcium channels, and so when they are depolarized, they open up and allow calcium to flood into that cardiac or to that muscle cell. There's also some beta adrenoreceptors. In fact, if you have parents or grandparents that take blood pressure medications, often they're taking calcium channel blockers or beta blockers, all right? And so that's where this terminology comes from, because your heart tissue is also affected by the same process. Okay, so depolarization due to that sodium channels opening affects the T-tubules. It opens up calcium ion channels here, and they flood the intercellular matrix, and that's important, okay? So calcium's the key here. So now we're looking at a close-up view of the actin-myosin complex. So remember, in the sarcomere, I've got the thick structure called myosin, and it's got that thickening in the middle that creates the M line, and also have these like thinner molecules called actin. And actin, like most proteins, is actually a complex of multiple protein subunits, okay? So you can see the actin is represented here by the circular alpha helical structure that underlies this. And then there's another molecule here, this long alpha helical protein that wraps around it called tropomyosin. Tropomyosin's a long word. This is a long protein that wraps around here. There's another little protein called the troponin. It's a globular protein, and it's important when it comes to actin, when it comes to this initiation events. One of the new things you'll see here are these myosin cross bridges, all right? So guess what the myosin cross bridges want to do? Well, they want to connect with actin, but they can't. There are little active sites on the actin molecule that are currently blocked by the tropomyosin troponin complex. You can't see them here, but you can see them here. Now, I know this is a kind of a faraway view, but when calcium enters the intracellular matrix, it actually binds the molecule troponin. All right, it binds that little fat globular protein. That causes a conformational change. So suddenly, tropomyosin and troponin shift their positions to reveal active sites on the actin molecule. All right, so here's the active sites, and these myosin cross bridges are immediately will form a bond with those active sites. All right, notice that myosin cross bridges are also associated with the molecule ADP plus PI, which really represents adenosine diphosphate with its inorganic phosphate ion. Now, when ADP and when this binding occurs, ADP and PI split, and they cause a change they allow binding, and then that causes this cross bridge to actually cock and move. And so it moves with respect to the myosin fibril, and that starts the initiation. ADP and PI come off, new ATP comes in and binds, and it causes that myosin cross bridge to break. And it's subsequently turned back into ADP and PI, and it causes the original myosin cross bridge to return to its original position. I know this is kind of hard to get through here, and it is complicated. You do have to rehearse it, and I'll put together a thing for you to do. But step by step, right? Calcium comes in, binds troponin, causes tropomyosin to shift. I've got the myosin cross bridge form. ADP and PI dissociate. It causes the bridge to move. ATP comes in, the bridges break, and then that ratcheting process happens again and again. All right? Me talking about it's not that helpful, but I will show you. I will give you some exercises to repeat the content. So in short, so remember that we have multiple sarcomeres in a muscle fiber and multiple myofibrils within the muscle cell. So when actin and myosin start that cross bridge formation, those cells ratchet up, and it happens in multiple sarcomeres at once, and this causes the contraction of the muscle fiber or muscle cell. Now, once you have contraction, you have to have relaxation, and that's the role of acetylcholinesterase, all right? So remember, acetylcholine is the neurotransmitter. Acetylcholinesterase is this enzyme that breaks down acetylcholine. Okay, so at some point you want the muscle to relax, and so acetylcholinesterase comes in, breaks down acetylcholine, and allows it to be released. The sodium ion channels in the motor end plate stop, the calcium flow ends, and then actually calcium is taken back up by the sarcoplasmic reticulum to be stored in the cisternae there. There's some inhibitory motor neurons that are associated with this again, but that's kind of complicated. Okay, last but not least are neurotoxins, all right? So yesterday I told you a little story about my dog Petite eating rat poison, and of course the agent in rat poison is strychnine. Strychnine comes from the poison nut tree, and it is an acetylcholine and glycine antagonist. So it actually affects acetylcholine, which is excitatory, and glycine, which is part of this inhibitory, relaxation phase, all right? And so it's an antagonist, all right? When we talk about drugs, we talk about agonists and antagonists, right? An antagonist, prevents you from doing what you want to do, right? If somebody's antagonizing you. An agonist, on the other hand, is the opposite of that. That actually sort of does what you are going to do. So the acetylcholine and glycine antagonists, it stops the effects of acetylcholine. It stops the inhibitory effects. So you get this like spastic paralysis, right? So I remember I told you about Petites, sort of spazzing out there on the ground. And of course, we use this as a rodenticide, right, for killing rats. Then there's the poison dart frog, right? So the poison dart frogs actually secrete this batrachotoxin on their skin, and the natives down in South America, where these things are found, can use these for hunting, right? So they take the poison dart frog and they squeeze it, make sure not to get it on themselves, but they'll put it on their arrows, and then they can hunt monkeys with their blow darts, right? So they have blow guns, and they can hit a monkey 80 feet in the air in his tree, and the monkey gets hit with the dart, and he kind of goes to sleep. He gets total involvement of a flaccid paralysis. The monkey will fall out of the tree and drop to the ground. The guy can pick it up. He's not dead yet, but he'll slowly suffocate due to the effects of the batrachotoxin, right? So it's a sodium ion channel agonist, right? It stops the sodium ions from moving in to allow that depolarization event, so it stops contraction. Now, Clostridium is a soil bacteria, and there's a couple variants that call it that make neurotoxins as well. One's tetani, and it's a GABA glycine antagonist, right? So I said that there are these neurons that are associated with the relaxation of the muscular contractions. These are inhibitory motor neurons, and GABA and glycine are these inhibitory neurotransmitters. So they block the effects, and you end up with lockjaw. So you can see this patient here with lockjaw. So you end up with tetanic contractions. That's a spasm that doesn't stop, right? Your muscles lock up. On the other hand, I have the Clostridium botulinum, and it blocks acetylcholinesterase vesicle fusion, right? So you get flaccid paralysis with this, and so the Clostridium spores are in the soil, but the way you know you can get botulism is with the canned goods. Tetani is introduced by cuts to your skin. So if you ever get a bad cut, you'll get a tetanus booster shot to inhibit the effects. But botulism comes in canned goods. So if you ever see a big swollen can, don't eat what's inside of it. Last but not least is this Novichok, which is a nerve agent, right? All over the world, governments have created nerve agents, and this one's an extremely powerful one. It's an acetylcholinesterase inhibitor, so it just stops the effects of acetylcholinesterase. So acetylcholine binds, sodium channels open up, and they never close. They're irreversibly bound open, and so you suffocate to death, or you have a heart attack. All right, well, that's a lot. I'll create an opportunity for you to rehearse the content, and I'll see you next time when we talk about sources of energy in muscle.